Hey guys, how you going? Welcome to Biology with Mr. V. This is the first video in the topic for DNA and proteins, which is the first topic for the year. So this is your very first video for the year. So welcome. Hopefully you've had a good start to the year. I won't hold up any longer and let's get started. So first of all, straight away you'll see just the topic outline and some of the key sub headings within this topic. So DNA structure, DNA and protein synthesis, protein structure and function and how that sort of works. Enzymes are a type of protein, so they're just a natural bigger heading within this unit. And then you have gene expression, mutations, and then biotech. So let's get started. The first thing you'll see in every single video is this. So you'll see key skills and understanding. And so the skills are highlighted in yellow and they're the things that they're like actions. They're the things that you'll actually have to be able to do. So just in context here, let's go with this one right here. Describe the structural properties of the DNA molecule, including nucleotide composition and pairing and such from there, right? You can see how that's a key skill versus understandings, which is things like DNA stores and transmits genetic information. It functions in the same way in all living things. That sentence doesn't necessarily describe something you have to do, whereas the yellow sentences are describing things where you will actually have to be able to do something with that knowledge. So they'll always be highlighted in the yellow. Okay, so I'll give you a second to read that and we'll just get straight on into it. So hopefully everyone's feeling a little bit, everyone's feeling a bit nostalgic because we're going to go back to some Year 10 science here, and this is DNA structure. So this is where DNA, we're talking about what it looks like and what are the actual chemical compositions within it. So there's a little bit of chemistry involved here. So if you are doing that, this sort of helps in that area, and chemistry will definitely come across little bits here and there in biology, but just in DNA structure, we typically call it a double helix structure. So you guys are probably used to that, where it's like a ladder that's simply just been twisted. And so that double helix structure is the overall structure of DNA, but within that, we have these other little bits and pieces. So the first basic unit of DNA that you have to understand is a nucleotide, right? Highlighted there in bold. A nucleotide, if you want to look at this image here, is the best way to describe it. A nucleotide consists of three parts. So you have a phosphate, a sugar group, and then it's connected to a base. And so when you look at it in the context of the DNA molecule in this image here, you can see how a phosphate, a sugar, and a base are constantly being repeated down a lane. So there's a nucleotide is added on to it, is like a building block. It's like a Lego brick, and you just add on more and more and more, and that's what DNA is made of. Now, there are four possible nucleotides, and that's because there are four possible organic bases, right? Adenine, thymine, A and T, and then cytosine and guanine, G and C, right? So within a nucleotide, the base that's attached is completely at random. So it could be a C, T, G, or an A, right? Adenine, thymine, cytosine, and guanine, okay? When they're inside the DNA structure, they have a corresponding base pair, partner, I should say. So for instance, an A will only ever, or adenine will only ever be able to bind to thymine, right? A thymine and vice versa. A thymine will only be able to bind to adenine. Cytosine and guanine will never be able to bond to an A or a T. Cytosine and guanine are complementary to each other. They'll bind to each other as well. So if you can see this image here, ATGC, THCG, they're constantly being repeated. Now, hopefully this is ringing some bells back to Year 10 science. We go, oh, I remember that ATCG, right? That's a really big part of DNA structure in that it is very, very specific. And so when we're talking about DNA structure and design, the main things you need to take away are it consists of nucleotides, and there are four different types of nucleotides, and then they're defined by what sort of base is attached to it. So whether it's an adenine, thymine, guanine, or cytosine, okay? Now, look at the types of bonds is actually quite important in DNA structure. So bases are bonded by weak hydrogen bonds. Now, you might think, what's the point in that? But it's actually one of the positives, it's one of the benefits of DNA. It's why it makes DNA such a good way to store genetic information, and so we can access it, because if they were held together in really strong pairs, right? Like if these bases here were held together in really strong bonds, if we wanted to separate them and use the information in the DNA, it'd be really, really hard. You'd need a lot of energy. Whereas weak hydrogen bonds means we can pull them apart easily and they'll just zip back together again quite nicely. And this is what makes DNA such a good structure to store information in, because it's easy to break apart. And that's genuinely one of the reasons that you can list DNA structure as a positive to store information is because it's got weak hydrogen bonds holding the bases together. This makes it really easy to use the information within the DNA. And we'll get to how the information in DNA is used later on. Okay. And again, inside DNA, well, not inside DNA here, I'm just talking about these last three: DNA, genes, and chromosomes. So DNA is the double helix structure, the ladder that's been twisted. A gene is a small section of DNA, and a gene is actually the coding portion. So a gene is made up of, let's say, for an example, very crude example here, 100 base pairs. So as in like 100 of these. That section would actually code for something that the body would need. So when we say gene, we're talking about a specific section of DNA that is actually going to code for something important. And genes do code for proteins. Okay. And then chromosomes are the last one. So chromosomes is a little bit different. Chromosomes, I think the next image will give me a bit of help here. Chromosome is typically looking like this. And a chromosome is made of DNA and genes. Because genes within DNA double ladder helix, what happens is when it wants to form a chromosome, and there's a specific point in time where it does this, what happens is the DNA starts to condense and coil, and it coils around these things called histones. You can see in this little image here. A histone is a protein, and what happens is the DNA essentially is like a shoestring, and it just wraps around it, and then it wraps around this histone and forms these nucleosomes, and then a whole bunch of nucleosomes get packed down together. It gets really, really squished, and then eventually you end up with a chromosome. And now the only time we ever really, the DNA wants to form chromosomes is when we're looking at cell division. We're looking at dividing a cell into two new cells, and that's just to make sure that the transmission of information is done correctly, okay? So again, this slide here does talk about a couple of things. So DNA is double helix structure. A gene is a small segment of DNA that codes for a protein or gene product. So again, get that in your heads. A gene actually will code for something, whether it's a protein or another gene type product. Okay. DNA forms chromosomes by condensing and coiling around proteins called histones. That's quite key there as well, right? The only point in time that we can see DNA within a cell is when it's in the chromosome-like structure. And that actually makes sense, because when you think about it, if DNA is uncondensed and is in its shoestring structure, like its double helix structure, and it's just stretched out, it's really, really thin. It's really, really hard to see. Whereas if you condense it right down and pack it all in together, it actually should become a bit more visible because it will become bulkier. Okay. Moving on. Chromosome structure. This is a big one. This is a big one where people will just really struggle with grasping. So I'm going to try and go through slowly, and you'll probably want to pause and replay this part of the video over and over again, which is totally normal, right? And you might want to ask follow-up questions in class or with whoever you are discussing this with, but I'll just go slow, okay? This image, I'm going to focus on this image. I'm going to ignore this one for the time being. These two images use red and blue, and that's to symbol paternal and maternal DNA. So in this example here, it hasn't been listed, so I'm going to do it myself. I'm going to say that the maternal DNA is red, which is maternal meaning mum, mother, DNA from the mother's side. So maternal DNA is red, and the paternal DNA is blue. And so inside of us, like inside of our regular cells, our normal cells, right? All of our cells have a full set of DNA. And so, for instance, we have 23 pairs of chromosomes. And so at each pair, right, we should typically have one side of paternal DNA and one side of maternal DNA. And that's because our genetic makeup is half our mothers, half our fathers. And so in this image here, we're using red and blue to show that, okay? When they're combined, right, and they form this X-like structure, we give them names. So when it's two blues connected together, so two lots of paternal chromosome. So essentially what you're looking at here is, this is sister chromatids, because they're the same color, so they're the same origin, so paternal DNA, and they're going to be the same chromosome. Let's call it chromosome number 1, right? Chromosome number 1 of the dad, and it's exactly the same on this side. So it's two copies of the same, and it's from the same parent, so we call them sister chromatids, right? Another thing we can look at here is, if we have it in an X like this, just like it says here, you have it in an X where it's two of the same color, so two maternal DNA two maternal chromosomes here connected together, the exact same, we would call that a chromosome. And then if they were to separate into an individual strand like this here, we could call this a chromosome because it is now separated from this structure. So it is a little bit confusing, but if you just go with it, as in like, if it's in an X and they are together, we can call the whole image a chromosome. If it's by itself, like a singular worm, we can call that a chromosome. When they're connected like this together, we would call each one, like in this image here, a sister chromatid. So on this red one here, that's one sister chromatid, and this is another sister chromatid, and that's because they're connected together. Now, what happens in our cells that aren't dividing, just functioning as per normal, our cells will have homologous chromosomes, and that's where we have one copy of paternal and one copy of maternal of the same chromosome, right? So for instance, on this chromosome here, let's call it chromosome number 1 from the dad, would be eye color, right? And we have the exact same chromosome from our mother, which also have eye color in there, and these two combining together will give us what we look like, right? So we call this a homologous chromosome when we have one of each, not when we have one of the same, okay? So I know that's confusing. We'll definitely go through this more together, but this is a really good way to think about it, okay? Moving on, we're looking at chromosome differences in prokaryotes and eukaryotes, okay? So prokaryotes, hopefully you do remember from year 11 and that they're really, really simple. Prokaryotes are cells that are super simple, as we can see here. They do not have any membrane-bound organelles. They're very, very basic organisms, right? They do have a cell wall, they do have a cell membrane, but they lack a lot of the internal structure that a eukaryote has. And so when we're thinking about chromosomes within a prokaryote, it's the same thing. A prokaryote only ever has one chromosome, right? And it looks somewhat like this, okay? One chromosome and it's circular, right? We always have to think of chromosomes are circular in prokaryotes. And that's one of the key differences, because if we go back an image, these are eukaryotic chromosomes. They're linear, they're in long forms. So that's a very, very key difference between them, in that in prokaryotes, a chromosome is circular, and we typically say they're found in the cytosol, which is another part of the internal liquid of a cell, right? Whereas in a eukaryote, they're linear, right? We call chromosomes linear in a eukaryote, and they're found in the cytoplasm, okay? Now, I won't get into this one. I'll talk about this a little bit later on, but this is basically talking about DNA found within chloroplasts and mitochondria and how this resembles an evolutionary event, okay? Now, first key checkpoint. So this is sort of a new heading within the same sort of realm of basic DNA structure. This is DNA replication, okay? So let's get straight on into it. We'll just make this a nice long video. This one is based on DNA replication. So if you don't want this part, you can just end the video here. But if you want to keep going, let's keep going. This is the next key concept of DNA replications, okay? So DNA replication, we're going to think about what the outcome is and when we want it to occur. So DNA replication takes a set of DNA, so one double helix structure inside of a normal cell, and creates two copies. So two fully preserved, fully functioning sets of DNA. And typically we would want this for when we're creating two new cells. Because what will happen is we have one cell, we want it to divide into two. We need both of those new cells to have a full set of DNA. So we have to go through this process first, okay? So just reading off here for a second, DNA replication is the process where a double-stranded DNA is split and forms two copies. This occurs in both prokaryotes and eukaryotes. Okay, so this process of replication will occur in both prokaryotes and eukaryotes because both those types of cells divide and make new cells, and so it's important that they have full sets of DNA. Okay, so DNA replication occurs in eukaryotes, and this occurs before mitosis, meiosis, whereas in prokaryotes, it occurs before binary fission. And if you remember from year 11, year 10-ish, this is where, that's the difference between how a prokaryote divides and a eukaryote divides. A eukaryote divides its cell and makes two new ones using mitosis. Prokaryotes use binary fission. Now, one of the key things you need to understand about DNA replication is the process is described as being semi-conservative, because what ends up happening is, if I look here, this is a good image for it. if I look here, I'm just going to move myself out of the way for a second. If I look here, this is the starting point, this blue strand of DNA. This is the original copy as it is. What happens is it's unzipped and then two new strands are formed. That's where these gold parts are coming in. So you start with, the starting of the point is one strand of DNA. The end point is two fully functioning, two full sets of DNA as it looks just like this. And we call it semi-conservative because half of each of the new strands contains half of the originals. Because what happens is, as it splits, the two sections that are being split apart serve as like templates. And what happens is, as one side is copied, a new strand is synthesized onto it, and you end up with these two here. So they both have a blue portion from the original. So it's half of the original, and it's conserving it because it's reusing it again. So when you think of semi-conservative, you think of half is being used again, right? Half is being used in the new copy. It's like being recycled, essentially. So that's this image here showing you that the original is a blue strand. It's unzipped. As it's unzipped, the two loose strands serve as a template. The gold strands are then synthesized onto them. So the new ones are synthesized onto them. And as it goes up, it works its way up and builds more and more and more until eventually you have two full sets of DNA using half blue strands on the new sets as the template. Okay. So I'll just go back over here. Okay. How the process sort of works. This is a step by step. Okay. So there's lots of things called enzymes used in here, and enzymes are key parts of how our bodies function. They're key little macromolecules that support us functioning. Proteins do a lot for us, and the enzymes are a type of protein. And typically an enzyme is recognizable because it ends in the letters ASE. So on this image here, you can see helicase, that's our first one, and ASE, and then the next protein or next enzyme is DNA polymerase, and it ends in ASE. And so typically the enzyme names give us the function. So for instance, helicase is the enzyme that unzips the DNA molecule. And what happens is it provides the two loose strands that can now be used to synthesize. As you can see here, as the helicase unzips it, the DNA is synthesized onto the original strands, forming new ones, right? That's the darker blue lines along these images. Okay, so what happens is helicase unzips, and then the other enzyme that supports it is DNA polymerase. Now, DNA polymerase binds onto the unzipped single strands of DNA. So it's sitting here attached just like this. And what it does is it actually attaches to the strand and then binds the complementary free-floating nucleotides within the cell into its specific point. So essentially what it's doing is binds on, and it reads it. It goes, all right, that green one is loose. Let's call it a cytosine. I need to find me a free-floating guanine nucleotide and attach it to keep building it. So it needs to find the correct Lego block to put it in the right spot. And so that's what DNA polymerase does. It's responsible for ensuring that the DNA that's being synthesized is synthesized correctly. It's getting the correct nucleotide and putting it in the correct spot. Okay, and now free-floating nucleotides are found in the cell. There's heaps of them. They're always built. They're ready to go. And the DNA polymerase just makes sure the correct one is attached to the correct base on the template strands. Okay, so DNA polymerase, because there's two sides of it, goes in two different directions. So DNA on the leading strand is synthesized in this direction, so it can just follow on with it, whereas the the template strand is synthesized, it's called the lagging strand, is synthesized in little bits and pieces. So you can see how here, this DNA polymerase has just started here and it's going to move this way, leaving this part. So what will happen is another DNA polymerase will come along and bind somewhere down the chain and then fill in this gap. So one strand is called the lagging strand, and that's the template strand of the DNA. And it's synthesized in this direction, going away, so it's sort of lagging, whereas the leading strand is synthesized leading towards the helicase enzyme. So it's constantly just built in one nice smooth chain, whereas the lagging strand is built in little bits and pieces. And we can go over that in class. Okay. Moving on. after the DNA polymerase is all finished and the process is completed, the DNA is just fully zipped back up again. And we use the word zip to describe it. That's one way to describe it. Another way to describe, for instance, the helicase separating it is unbinding or breaking the weak hydrogen bonds between them. And this is where that weak hydrogen bond interaction, like why it's important that they're bound together by weak bonds, comes in, because this means the helicase enzyme can unzip the DNA molecule quite easily. It doesn't need a lot of energy at all. It can simply unbind the two strands easily because the weak hydrogen bonds don't really hold the DNA together all that well. So that is a benefit of the DNA structure. Moving on, that's our overall outcome. Right, that's where this video ends here, guys. That's DNA replication. Again, we'll go over it in class together. I'll just move myself for this last part here. Right, we'll go over this in class together and go through some questions. But overall, we've just gone through DNA structure, what it sort of looks like, and DNA replication. All right, I look forward to seeing you next video. See you later, guys. Bye.